Uh, I'm glad to be here, see you guys all at Montreal, and have the opportunity to uh, talk my project, the model named Forseti, which is a mechanistic model that can predict splicing status of the single-cell RNA-seq reads. The single-cell RNA-seq technology has become one of the most popular tools for the transcriptome-wise analysis, and which also revolutionized our understanding of the cellular heterogeneity and the rare cell type identification. And most analysis utilize the count matrix that doesn't distinguish between different splicing status of the reads. But the recent research have highlighted that the information captured from the unspliced reads can bring novel biological insights for downstream analysis. For example, the VeloCyto, the SC-Velo, those tools were developed to estimate the RNA velocity, to explore the cellular dynamic, and they also open up new ways for the cell differentiation and disease progression. However, currently determining the splicing status for the single-cell RNA-seq reads remains a unsolved problem, and currently the standard method, they classify the reads that map to the fully map to or partially map to the intronic region, classify this reads to the unspliced reads, and for reads that only map to the exonic region was classified to the spliced reads. However, for reads that map to within one exon, they can actually originate from unspliced transcript or the spliced transcript. So for those reads, instead of classifying them as a spliced reads, they are actually ambiguous. So there's a strong preference for classifying this exonic reads as spliced. Furthermore, according to the statistical data, we have 20 to 40% UMIs or reads that are actually ambiguous. So there's a pressing need for a sophisticated model to accurately assign the splicing status for those reads, especially for those exonic reads. And here you might ask, like, can we find the actual splicing status for those ambiguous reads or ambiguous UMI? And what information did we miss before? So here, we can take a quick look about the single-cell 3' sequencing workflow, and let's see what information we can find, what are the hidden message we can find here. So during the cell partitioning, the single cells are encapsulated into gel beads in emulsion, also called a gem. So in each valid gem, it should contain one single cell and one gel bead. And when we zoom in each gel bead, on its surface, there are millions of oligoprimer. And each primer should contain a read one sequence, that cell barcode, the UMI, and a poly-DT primer. And inside each gem, the oligoprimers are released and cells are lysed. Then the poly-DT region on the primer will target the 3' poly A tail of the mRNA. And if there are regions on the mRNA that contain any poly A stretch, it will be primed as well. So here, this is the first hidden message, that the priming site we are looking for should contain a poly A stretch, and this priming sites can either come from the poly A tail or some internal poly A stretch. And after that, the mRNA is reverse transcribed to the double-strand cDNA, and the pooled cDNA will be processed in bulk. And there are 2 rounds of the PCR, and for the first round, the full-length cDNA will be amplified. And then the full-length cDNA will be randomly fragmented to the cDNA fragments. So for those CDN fragments, even though they come from the full-length cDNA with various with various lengths, most of the CDN fragments here are 300 to 400 base pair long. So this is the second hidden message we found from the text platform. So here with these two hidden messages, we can actually infer the priming window downstream of the read along the transcript. And ideally, the inferred priming window, can you see my mouse? Okay, so here, ideally, the inferred priming window should not be too far or too close to the observed read. And we hope that the inferred priming window have a relative higher binding affinity to the oligo-DT primer. And assume that we have two models that can help us to calculate the fragment distribution, the probability, and have models to help us calculate the binding affinity. Then we can identify the most possible transcript that give rise to the observed read. And here we introduced Forseti, which contains two components. And here I'm going to introduce the first one, the fragment distribution. And here the basic idea is that we use the pair-end dataset to compute the fragment distribution and fit spline model for it. And I believe most people are much more familiar with the single-end dataset, the single-end sequencing where the read one is much shorter, which only contains the technical region. It has the text barcode and UMI. And in the read two, it has the biological meaningful sequence. And the pair-end sequencing is also available on 10x. So here, both ends of the CD insert was sequenced at the same cycle length. So the example here is 150 cycles. And in the pair-end dataset, the read one contains the technical region, have the barcode UMI, as well as the biological meaningful sequence. So here, both ends of the CD inserts are alignable. And then we can use the mapping position of both biological read one and the read two to compute the fragment length. And this fragment length is also the distance between the read two and the priming window. Then we use a cubic spline model to fit this distribution. And with this model, we can compute the probability that we observing a fragment with the given length. And then the second component is a oligo-DT primer binding affinity model. So here, we extract the priming window from the read one. And for this window, we know that this is the region that can be primed by the oligo-DT primer. And we will consider this is the positive class. And in particular, this 30-mer window can be extracted based on the position of the read one mapping to the reference genome. And then we also randomly extract 30-mer window from other positions. And this 30-mer windows may not be primed by the oligo primer, and we consider them as the negative class. And then we use both classes to train the multi-layer perceptron model. And then we can use this model to calculate the likelihood that we classify a 30-mer window to the positive class. And then we use this likelihood to infer the binding affinity of the oligo-DT primer to the 30-mer window. And now we have both models ready, and those models allow us to compute the probability that the transcript generates the observed read. So here for this transcript, we find all possible priming window downstream of the observed read. And for each priming window, we first compute the distance between the window to the start of the read. And this is also the fragment length we defined before. And then we use this distribution in our first model to calculate the first probability, the fragment length probability. And then we infer the binding affinity of this priming window to the oligo-DT primer. Then here we use the joint probability to calculate the likelihood that this transcript gave rise to this observed read at this priming site. Okay, and then the final probability that this transcript gave rise to the reads which align at the position of xt is the highest probability at the best priming site. And then now here, this is the last step that we can calculate the probability of being a spliced read or a unspliced read. So we might have multiple transcripts for the both splice status. For example, here we have 2 unspliced transcripts, and we only return, we only keep the highest probability, like 0.5 here for the unspliced transcript, and we use 0.1 for the spliced transcript. And here, the probability of being a splice read is the highest probability from splice read, which is 0.1 over the sum of the highest probability from both splice status. And here is 0.1 plus 0.5. And then we got the probability for the splicing status. And if there's a tie between the splice and unspliced site, unspliced status, which means the final probability here is 0.5, we assign the reads as ambiguous reads. Otherwise, they will either classify to the spliced or unspliced reads. And then we evaluate the performance of Forseti on 2 experimental data sets from mouse and human. And we plot our ROC curve and we calculate the AUC score. And the mouse and human data set got 0.89 and 0.85 separately. And those high AUC scores suggest that the Forseti have a pretty good prediction power. And to summarize, here the Forseti model contains 2 components. The first one, we utilize the pair-end dataset to calculate the fragment length, and then we fit a model for its distribution. And then we extract the priming window from the data, and we have a binding affinity model. Then we combine both models, and we use the joint probability to calculate the likelihood that the transcript gave rise to the observed read, and then we determine the splicing status of the read. And then I want to say thank you to my advisor and all my lovely members, and I'm happy to take questions here.
So we have time for a couple of questions.
Hello.
Hello.
Thank you very much for your nice presentation. So we are now more and more using long reads to identify splicing patterns. I was wondering, and one of the things that we are finding using the long reads is that there are a lot of new transcripts that are not in the reference annotations. So I was wondering how this kind of uncertainty on the actual composition of the transcriptome affects your algorithm.
Sorry, can you repeat your question?
So the fact that they may be transcripts that are not in the reference annotation. So how is this affecting your algorithm?
Affecting?
affecting? So how does impacts? So my question is, under the assumption that there might be a transcript that you don't know at all because they are not in any reference.
You mean
My question
like
is,
the novel
how is this?
isoforms?
Yeah, unknown isoforms. How this can affect your algorithm?
Yeah, I think this is a great question because the reference we use, we use like splicing and unspliced reference, and I think this we just based on the known transcript, like the discovered transcript. If they are like the novel transcript and they are like have a big proportion of that, maybe we won't able to find. Yeah, we won't able to find the status for that.
Thanks.
Hi, so nice
Hello.
talk. So I was wondering how does your model sort of cope with the fragment length distribution? Let's say, for instance, you had a wider fragment length, and what's the implications of your predictability?
You mean how do I get the fragment distribution?
So let's say, I mean, fragment distributions can be a little bit varying in width.
Yeah.
How, like, if you could just comment on how it affects your model, like if you had a wider one compared to a more narrow one.
So I collect the data from the 10x. So your question is that like a generalized one can apply that applicable to different data set? Is that your question?
Maybe I misunderstood, but I just wondered, like, with more uncertainty in data, like the length, because you talked about the length, the fragment lengths and use to predict something. So with more uncertainty, like variability in the fragment length, maybe there's some, how does that affect your model or predictions? Maybe we can take it offline.
Okay. Yeah, I think your question is like some outliers with different, more different fragmenters. Is that right?
Yes. Yeah, we can talk about it offline.
Okay. Okay. I think I can provide more details of this. So we have a like a regional distribution. They have some fragment learns like exceed the 1,000 base pair, and we for now we just ignore the fragment learns distribution like over 1,000, and now we focus on the distribution like from 0 to 1,000. Yeah, and also I also want to mention that if there's like data that side from other platform and they may have a different fragment distribution, and to use our model, it can like easily replace with a new fragment distribution, and also combined with the rest part of the whole model. Okay, thank you.
Okay. Well,